Welcome, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Maxcyte, I'd like to welcome you to Synergizing Transient and Stable Protein Expression for Accelerated Biotherapeutic Development. My name is Elizabeth Lamb, and I'll be the host and the moderator for today's event. At this time, I'd like to introduce our presenter for today. She is Dr. Krista Stager, PhD, with Maxcyte. Welcome, Krista. The presenter ball is yours.
Thanks, Elizabeth. I'd like to start by welcoming all of you to the webinar. As Elizabeth said, my name is Krista Stager, and I'm a scientist here at Maxcyte. Today, I'll be discussing the changing role of transient transfection within biotherapeutic development pipelines, and specifically how advances in transient protein production are driving this evolution. I wanted to start out here with an outline of what I hope to cover in today's webinar. I'll begin with a historic perspective of how transient protein production has been used during biotherapeutic development, and discuss several of the obstacles that have limited its use. Before discussing the expanded use of transient transfection, I'll briefly introduce Maxcyte Electroporation for those of you who may not be familiar with its capabilities. Next, I'll focus on how the high level of performance and scalability of Maxcyte technology is expanding the footprint of transient transfection, ultimately with the goal of positively impacting development timelines and costs. I will present data illustrating gram-scale production of recombinant proteins, including antibodies and antibody-like molecules from a variety of CHO cell lines using transient transfection. Lastly, I'll discuss how Maxcyte transfection seamlessly segues into stable pool and stable cell line generation, shortening this phase of development as well. During the beginning stages of biotherapeutic discovery, hundreds of candidates are being evaluated to identify the more promising candidates based on binding affinity, biological activity, or other relevant metrics. Although the number of candidates is high at this stage, the assays typically being performed require only lower amounts of the protein. Transient transfection was commonly used to rapidly produce the micro to milligram quantities of proteins that were needed. Historically, however, CHO cell transfection efficiencies and cell viabilities were problematic, leading to the use of HEK cells as the avenue for transient production. CHO-based stable cell lines have been and actually continue to be the ultimate means of manufacturing the therapeutic antibodies, in part due to regulatory ease. Thus, at some point during the development process, stable cell lines must be generated. The process, as many of you most likely know, is generally lengthy, very labor-intensive, and can be costly, leaving researchers faced with the challenge of deciding when during the development that effort is undertaken. If companies choose to generate stable cell lines very early in the process, it requires the creation of large numbers of stable cell lines, most of which in fact represent candidates that will most likely fail during later stage development. If a company elects to delay the investment in stable cell line generation until the number of candidates is reduced, much of the early development may be in a cell line, such as HEK cells, that differ from the manufacturing cell line. By beginning development in HEK cells, differences in post-translational modifications and in protein stability relative to proteins produced in CHO cells may confound interpretation of bioefficacy data. In addition, many biomanufacturing challenges may not become evident until late in the development process. Lastly, if the stable cell line generation is delayed until the later phases, the process to get the low quantity of protein produced transiently may limit the degree of antibody characterization that can be performed. So researchers, again, face a delicate balance between the cost of resources required to produce a large number of stable cell lines early in development versus the relevance of the data being generated and the amount of protein you can produce. Maxcyte transient transfection is scalable electroporation-based cell loading technology that can rapidly generate milligram to gram quantities of proteins in the cell line of choice, including CHO cells. This reduces risks by enabling earlier and more detailed analysis of antibody candidates that have the correct post-translational modifications, all prior to investing in the generation of cell lines. Additionally, the high performance of Maxcyte Electroporation can significantly reduce the time needed for cell recovery during selection and create stable pools enriched for high producers. In this way, Maxcyte Electroporation accelerates biotherapeutic development not only through high-titer transient protein production, but also by streamlining stable pool and stable cell line generation. Maxcyte Transfection can also significantly streamline development timelines by enabling parallel tracking of transient protein production for their characterization and generation of stable cell lines, thus eliminating any delays within the development process for migration to stable protein expression. In the next few slides, I'd like to give you a brief overview of Maxcyte's scalable electroporation technology. I'll present data demonstrating the platform's capabilities, including cell type flexibility and the flexibility in the type of proteins being produced. The high levels of transfection efficiency, cell viability, and the productivity of Maxcyte transfected cells will be highlighted. Additionally, I'll present data demonstrating Maxcyte's unmatched scalability that works with a high level of performance to greatly expand the role of transient transfection within biotherapeutic development. For those of you who are not familiar with Maxcyte or Maxcyte's technology, I'd like to give you a bit of a background about our company. Maxcyte has been in business for over 16 years. Our scalable electroporation technology was originally developed for cell therapy applications, and we're still very actively involved in the cell therapy arena. Maxcyte's technology offers a non-viral approach for modifying the function or the phenotype of primary cells and stem cells with the high levels of cell viability, transfection efficiency, and safety that are key in clinical applications. Our system is supported by a U.S. FDA master file that's been referenced in multiple clinical trials, including a number of recent studies involving the expression of chimeric antigen receptors in autologous T cells. The expertise that we developed in transfecting challenging primary cells on the clinical side of our business attracted the attention of many pharmaceutical and biotech companies that wanted to express drug targets such as GPCRs, ion channels, and kinases, as well as receptor molecules for cell-based assays. Large-scale transient transfection is much faster than making stable cell lines, and it allows these users to generate assays with primary cells and physiologically relevant cells. Multi-subunit proteins and potentially toxic proteins can be screened more easily using transient expression, and the cells can actually be aliquoted and cryopreserved following a large-scale electroporation, which allows users to run assays at their convenience. Now, the third application area where our technology has been widely adopted, and in fact, the field of use that's most relevant to today's webinar, is bioproduction. Maxcyte users can rapidly produce large quantities of recombinant proteins, including antibodies and antibody-like molecules that we'll discuss today, but also for viral vectors, vaccines, and other recombinant antigens in cells that provide for the correct post-translational modification. In addition, Maxcyte technology also facilitates the generation of stable pools and cell lines. This slide highlights key attributes of Maxcyte's transfection technology. First, with most cell types, we can achieve greater than 90% transfection efficiency while maintaining greater than 90% cell viability. Maxcyte's electroporation-based technology can transfect or co-transfect a wide variety of loading agents, including plasmid DNA, mRNA, siRNA, proteins, and even cell lysates. Temporary openings are introduced into the cell membrane by exposing the cells to a brief series of electrical pulses. Any type of molecule can be introduced into the cell using this process, and no adjustments to the electrical parameters are required to accommodate these different loading agents. The technology is compatible with a wide variety of cell lines, as well as primary cells and stem cells. For all of these cell types, a single chemically defined, animal component-free electroporation buffer is used. Maxcyte electroporation is extremely scalable, able to transfect from 500,000 to 200 billion cells. Smaller numbers of cells are transfected in a cuvette-style chamber via static electroporation. At this scale, the process takes only a few seconds. Larger numbers of cells can be transfected by flow electroporation using the same instrument with the same electroporation protocols. No reoptimization is needed. Both static and flow electroporations take place in sterile, single-use processing assemblies, so there's no risk of contamination from one transfection to the next. The studies presented throughout the remainder of the webinar were performed using one of two Maxcyte electroporation systems, either the Maxcyte STX or the Maxcyte VLX, both of which are cGMP compliant, they're ISO certified, and CE marked. One of the most unique features of the Maxcyte platform is its scalability. The Maxcyte STX, shown on the left, can transfect as few as 500,000 cells up to 20 billion, or 2 times 10 to the 10th cells in a single transfection. The different size processing assemblies, which are used with the STX, are shown on the lower left, including the cuvette-styled OC100 and OC400 chambers, the intermediate scale CL1.1, and the CL2 assembly used for flow electroporation. Again, all are single-use and disposable to maintain sterility. So in addition to the STX, Maxcyte also offers a larger scale instrument, the Maxcyte VLX, which can transfect up to 200 billion cells at one time. No reoptimization is required to scale up from low-volume static electroporation to flow electroporation on the STX, or during scale-up from the STX to the VLX system, enabling this single cell engineering platform to take you from R&D through to biomanufacturing. Both the Maxcyte STX and VLX come with a preloaded library of electroporation protocols that Maxcyte scientist has optimized for individual cell types. Users do not need to make any adjustments to the electrical parameters. They simply select the protocol optimized for their cell type from a drop-down menu in the instrument software. This slide shows our current list of optimized protocols for over 80 different cell types. Cells commonly used for bioproduction are highlighted in blue. Our list of cell type-specific protocols is continually expanding, but if your cell type of interest is not on the list, we also provide 10 optimization protocols ranked from low to high energy that should allow you to easily find conditions that provide good transfection efficiency and cell viability for your cell type. Here I'm showing you images of cells that we transfected with a GFP expression plasmid using the Maxcyte STX to give you a visual representation of the high levels of transfection efficiency and viability that can be achieved using Maxcyte electroporation. In this slide, I've specifically chosen cells that are used for bioproduction, including CHO cells, in which we routinely see greater than 95% transfection efficiency and cell viability. In addition to a variety of mammalian cell types, Maxcyte technology also provides highly efficient transfection of insect cells, such as SF9s. This can eliminate the need to generate baculovirus in order to introduce genes into insect cells, saving significant time and effort. As I just showed you, Maxcyte's technology can transfect CHO cells with nearly 100% efficiency while maintaining greater than 95% cell viability, which are much higher than what is typically achieved with other transfection methods. Our high levels of CHO transfection efficiency and cell viability translate into a high level of productivity. In the experiment on this slide, you can see that titers of more than 1 gram per liter were achieved following electroporation of CHO-S cells with a bicistronic plasmid encoding human IgG1 heavy and light chain proteins. 8 times 10 to the 7th cells were transfected at small scale using static electroporation in an OC400 processing assembly, and 1 times 10 to the 10th cells were transfected at large scale via flow electroporation in a CL2 processing assembly, both on the Maxcyte STX using identical electroporation protocols. Both small and large-scale transfection produced highly consistent antibody titers of over 1 gram per liter with comparable expression kinetics, highlighting the ease of scalability. For this particular antibody, using a batch-fed process with commercially available base media and feed supplements, we generated well over 3 grams of antibody from a single 3-liter shake flask via transient expression. I'll refer back to this gram per liter data a little later in the presentation when I touch on augmenting protein titers through culture process development. Before I move on, I also want to mention Maxcyte users can return their cells back into culture following transfection at higher cell densities. In this experiment, cells from the CL2 were transferred to a 3-liter shake flask at a density of 4 to 5 million cells per mil, which exceeds what is typically used following other transfection methods. In addition to simplifying cell culture logistics, the ability to culture cells at higher densities minimizes media consumption and simplifies downstream purification. The previous slide focused on the ability to move from small-scale static electroporation to large-scale flow electroporation on the Maxcyte STX without reoptimizing electroporation parameters. This slide demonstrates the platform scalability between the Maxcyte STX and the Maxcyte VLX. A side-by-side comparison in which 20 billion CHO cells were transfected with a human IgG1 expression plasmid using flow electroporation on either the STX or the Maxcyte VLX was performed by a current client. You can clearly see that cell viability and growth, as well as antibody production, were comparable over the 2-week period following electroporation for both instruments, again demonstrating the true scalability of Maxcyte electroporation. In this next section of the webinar, I would like to share how the attributes of Maxcyte electroporation that I just discussed, such as cell and protein type flexibility, and the high level of productivity, enable the expanded footprint of transient transfection within development pipelines. Data will illustrate the transfection of multiple CHO cell lines, the expression of multiple types of proteins, as well as the high quality of the proteins being produced. The last topic in this section that I will touch on is the high degree of similarity between transiently and stably produced proteins. For transient transfection to truly expand its roles, researchers must be confident that transiently produced proteins are representative of the final stably produced proteins and thus can be used as surrogates during development. The scalability studies that I shared in the last section of the webinar were all conducted using CHO-S cells. The next several slides will show you data for other commonly used CHO cell lines, including CHOZN cells, a zinc finger modified GS knockout cell line, which is highlighted in this slide. 3 independent IgG transfections of CHOZN cells were performed using the Maxcyte STX. Conditioned media were collected on days 5 through 8 post-electroporation and antibody titers determined. The graph on the left shows consistent antibody titers for the 3 independent transfections, with titers increasing throughout the post-transfection culture period. In addition to antibody titer analysis, day 8 samples from transfections number 1 and transfection number 3 were purified using protein A and examined via gel electrophoresis. Bands of the expected size for human IgG heavy and light chains are evident on a reducing gel for both protein A purified and unpurified samples. Also, no additional bands are evident in the purified samples, which are in lanes 3 and 4, indicating expression of high-quality antibody. The panel on the left of this next slide shows greater than 95% transfection efficiency for CHO K1SV cells following Maxcyte electroporation with a GFP plasmid. Customer data of side-by-side transfections with a human IgG expression plasmid comparing Maxcyte electroporation to chemical transfection of CHO K1SV cells are shown on the right side graph. Chemical transfection results in extremely low titers on day 5 post-transfection, with negligible titers on day 9 and 13 post-transfection. In contrast, Maxcyte transfected cells exhibited robust antibody expression throughout the 2-week culture period. Of note, the titer reached over 1 gram per liter in this experiment without any optimization of post-transfection culture conditions, demonstrating the high level of productivity of Maxcyte transfected cells. The customer study summarized here in this slide represents transfection of CHO-K1 cells with a non-antibody therapeutic protein using 4 different transient transfection methods, specifically Maxcyte Electroporation, a lipid-based reagent, a polymer-based method, and an alternative electroporation system. 3 independent transfections were performed using Maxcyte Electroporation and compared to 2 transfections using each of the other 3 methods. Conditioned media samples were taken on days 5, 6, and 9 post-transfection for titer determination. As you can see, Maxcyte Electroporation significantly outperformed all other transfection methods throughout the 9 days of culture. Of note, cultures of lipid or polymer transfected cells showed extremely low cell viability after 6 days in culture, and thus no titers were observed for those 2 methods at day 9 post-transfection. I also would like to share some data on transient expression of proteins other than conventional monoclonal antibodies. We just saw expression of a non-antibody therapeutic protein in the previous slide. Here in this study, we expressed a diabody using static electroporation of a bicistronic plasmid in CHO-S cells. By way of comparison, the client produced the same diabody in their laboratory using their previous lipid-based method. Samples from both transfections were purified by the client, quantified, and their quality analyzed through chromatography. Diabody titers following Maxcyte electroporation were 20 times greater than those following lipid-based transfection. Additionally, the Maxcyte-produced diabody showed in the lower right panel was of high quality with very low levels of aggregation compared with the client's previous transfection method shown in the lower left. I've shown you a few examples of the high level of productivity we achieve using different CHO cell lines in comparison to other transfection methods. These were just a glimpse of the many side-by-side method comparisons we have done both internally and at customer sites. This graph is simply meant to convey the point that for a wide spectrum of protein types, from full IgGs to small viral proteins, we find Maxcyte transfection well outperforms other transfection methods. I'd like to shift gears a little here and spend the next few slides illustrating the quality of proteins produced using Maxcyte electroporation and their similarities to stably produced proteins. In the study shown on this slide, the client had previously created a stable cell line to use as a reference. CHO cells were transiently transfected using small-scale Maxcyte electroporation with an IgG expression plasmid. Conditioned media from transiently and stably transfected cells were analyzed by reducing and non-reducing gel electrophoresis as well as isoelectric focusing. The analysis shows no detectable differences between stably and transiently produced proteins, with clean bands of the appropriate molecular weights visible. The next 2 slides are from an experiment in which we conducted 5 independent transient transfections of CHO-S cells using a human IgG construct. A stable cell line was generated from one of the 5 transfections. The 5 sets of cells were cultured with the same base media and feed supplements. Antibody was purified via protein A from the 4 transient transfection cultures and an identified high-producing stable clone. This gel illustrates the consistently high level of antibody quality and the high degree of similarity between antibodies stably and transiently produced. So it's well established that protein glycosylation varies depending on the cell background. Proteins produced via transient transfection in HEK cells have different patterns of glycosylation than those produced in CHO cells. In addition, it's known feed and media supplements can influence protein glycosylation. Our goal was to assess the impact on glycosylation of expressing a protein via transient CHO transfection versus stably from a cell line. The samples we examined for quality in the previous slide were also examined for glycosylation. Glycoform analysis of the resulting proteins showed consistent patterns of glycosylation among all 4 sets of transiently transfected cells. These data also demonstrate consistent glycosylation patterns between proteins produced transiently in CHO cells and the same protein expressed by a stable CHO cell line generated following Maxcyte electroporation. This highlights that if cells are maintained under consistent culture conditions, Maxcyte Transient Transfection allows users to produce high levels of protein early in the development that's highly representative of the final manufactured product. The data I've presented thus far have used unoptimized post-electroporation culture conditions. Multiple variables, both pre- and post-transfection, influence recombinant protein titers in transiently transfected cells. Of course, to maximize titers, it's essential to start with high transfection efficiency and high cell viability. That's where Maxcyte comes into the picture, providing a strong foundation to build on. Prior to transfection, choosing the right host cell line, maintaining healthy cells, and selection of the right expression vector will influence production levels. After transfection, efforts to monitor metabolites, waste products, dissolved oxygen levels, and pH while maintaining nutrient balance will further enhance titers. These activities fall under the umbrella of process development. In earlier slides, antibody titers of greater than 1 gram per liter were presented for cultures in which post-electroporation culture conditions had not been optimized. The data shown here illustrates how even a minimal amount of process development can significantly improve antibody titers. In this experiment, we transfected CHOS cells with the same antibody expression plasmid that we used in the earlier scalability data. Transfected cells were divided into 3 shake flasks where we tested different base media and feed supplements. Process 1 replicated the conditions from the previous experiment and again gave titers of around 1 gram per liter. However, you can see through a small degree of process development, we were able to push transient antibody titers in a CHO cell to nearly 3 grams per liter while still using commercially available base media and feed supplements. The capacity to produce protein titers approaching 3 grams per liter, combined with Maxcyte scalability, which you'll remember is up to 200 billion CHO cells in under 30 minutes, equates to the production of multiple grams of quality antibody within 2 weeks of a single transient transfection. I'd like to pose the question to you of what would you do when given the capacity to rapidly generate tens of grams of protein much earlier in the development process? What types of characterizations could you do to de-risk candidates prior to investing in stable cell line generation? I've been focusing on the benefits of Maxcyte Transient Transfection for delivering gram-level quantities of proteins that are representative of stably produced protein. Hopefully, this has inspired you to consider how this capability could expand the role of transient transfection in your biotherapeutic pipeline and reduce the need to quickly move to stable cell line generation. Ultimately, though, you need to stably express your therapeutic. So in the last section of the webinar, I'd like to share how Maxcyte electroporation offers significant advantages for rapidly generating stable pools and clonally derived stable cell lines. I will also discuss the extensive work we have done to establish the quality and robustness of the generated single-cell clones. A great deal of data has been shown for how the high levels of Maxcyte transfection efficiency and high cell viability positively impact transient protein production. But these attributes also impact stable pool generation by enabling more stringent selection protocols. Maxcyte transfected cells typically recover more quickly from selection compared to cells transfected via other methods. In this slide, customer data are summarized for 12 independent transfections. 4 performed via chemical transfection, which are shown in green, and 8 performed via Maxcyte electroporation, which are shown in red and blue. Half of the transfections for each method were for the expression of a full IgG, while the other half were for the expression of an Fc fragment. 2 of the 4 chemical transfections, specifically those transfected with an Fc expression plasmid, did not fully recover, with almost complete cell loss at day 16 post-transfection. Cells from all 8 Maxcyte transfections, so those expressing either the IgG or the Fc fragment, all recovered 8 days earlier than the 2 remaining chemically transfected cells. Again, this highlights the significant impact Maxcyte's high cell viability can have throughout all of the protein expression efforts. In the last set of experiments I'll share, we used Maxcyte transfection to generate stable cell lines through limiting dilution cloning and subsequently characterize the robustness of a high-producing clone. We began by applying selection pressure 1 day after electroporation of CHOZN cells with a bicistronic antibody plasmid. Within 2 weeks, cells recovered from selection and produced a stable pool. At that point, we initiated limiting dilution cloning, and at the end of 8 weeks, the highest producing clone was placed into production culture. After screening fewer than 500 clones, a relatively small number, we identified a high-producing clone, number 17, whose titers exceeded 3 grams per liter during culture scale-up, as can be seen in the upper left graph. In all 4 graphs on the left side of the slide, day 0 in the data set represents the start of the production run, which equates to only 6 weeks after the original electroporation. Through further development of culture conditions, clone 17 productivity reached just under 6 grams per liter. To assess the robustness of this clone, we created a cell bank at the end of the 6-week cell line development process, and then again following in an additional 30 and 60 days in culture. All 3 cell banks were thawed and passaged for 2 weeks, and then 18-day production cultures were seeded. Antibody titers and conditioned media samples from day 10, 14, 16, and 18 of the production cultures were determined. A less than 15% loss in productivity is detected between the first, second, and third cell banks, highlighting clone robustness. In addition to antibody titers, we examined the quality and glycosylation patterns of the 3 cell banks, which again represents a 60-day culture period. No loss in antibody quality is detected, nor is there a significant shift in glycoform abundance. Thus, Maxcyte electroporation represents a means to shorten the timeline to stable pool production, to enrich stable pools for high producers due to the increased selection pressure that can be applied, and to generate high-yield, robust, stable clones. Data during this webinar highlighted the ability of Maxcyte transient transfection to produce milligram to tens of grams of proteins within weeks rather than months of a single transient CHO cell transfection, which allows you to expand the characterization of candidates prior to investing in cell line development. Data also highlighted the ability to accelerate stable pool and stable cell line creation, further accelerating biotherapeutic development. If clients are looking for maximum compression of the development pipeline, it is possible to parallel track early and mid-stage development with stable cell line generations. Users can perform a single bulk transfection and use a portion of the transfected cells to generate transiently produced protein for generating analytical and activity data, and at the same time subject a portion of the same cell bulk transfection to selection, beginning the process of stable cell line generation. I wanted to conclude today's webinar by summarizing a few key take-home messages regarding Maxcyte's scalable transfection technology. First, Maxcyte's technology can transfect a wide range of mammalian cell lines, as well as primary cells and even insect cells. In particular, the Maxcyte system transfects CHO cells with high efficiency and high viability, allowing users to screen antibody candidates via transient expression in the same cell background that ultimately will be used for biomanufacturing. Furthermore, Maxcyte users are not restricted to working with a specific strain of CHO cells, and they are free to use any commercially available or proprietary media formulations and expression vectors. In addition to compatibility with many different cell types, Maxcyte's versatility extends to the molecules and products that can be expressed. These include secreted, membrane-bound, and intracellular proteins. As you've seen today, a variety of antibody and antibody-like molecules as well. The seamless scalability, high transfection efficiency, and high post-transfection cell viability that characterize Maxcyte's transfection process led to very high transient protein titers. Thus, it's possible to generate gram quantities of antibodies via transient expression in a CHO cell, often while culturing cells in shake flasks instead of cell bags or bioreactors. In cases where it's necessary to generate stable pools or stable clones, starting with Maxcyte Transfection often allows users to reduce timelines for pool generation and to reduce the number of clones that must be screened in order to identify a high expressor. These benefits again stem from the high levels of efficiency and viability that are characteristic of the Maxcyte transfection process. Additionally, it is possible to parallel early candidate characterization by generating analytical and activity data with a transiently produced product with stable cell line generation, further condensing the development timeline. The bottom line, though, is that all of these advantages add up to significant time and cost savings. At this time, I'd like to thank you for your time today. I hope you found this information useful, and I welcome any questions you may have.
Thank you very much, Dr. Stager. We do have several questions. Our first question, if I wanted to switch from another transfection method to Maxcyte, what optimization do I need to do?
As I showed, we've got an entire library preloaded under both the STX and the VLX for the electroporation protocols or the parameters that are optimized for specific cell types. So in fact, it's very easy to switch from another transient method by simply using those conditions that we've already optimized.
Okay, our next question, can I use this with wave bags?
Absolutely. We've had a number of clients that when they scale choose to culture cells post-transfection in wave bags or a number of other bioreactor types.
All right, perfect. Our third question, do you have an issue with plasmid size limitation?
Oh, great question. No, we do not. Sometimes if people choose to have a single expression construct where the IgG heavy and light chain are in tandem, even though the plasmid can be a bit larger, we do not have any problems whatsoever. In fact, our plasmid size can be as high as 35 kD. So people can choose and assess whether they'd like to use, again, a single plasmid with the heavy and light chains in tandem, or whether they choose to use two different expression constructs. Sometimes using two expression constructs, they'll find they can get higher expression, or in fact, they can play around with the ratios of the two expression plasmids to, again, try to optimize the final product.
All right, got it. Our next question, at what cell density do you electroporate cells?
Another good question. So I talked about the fact that post-transfection you can have higher cell densities, but in fact during the actual transfection process we find we can use higher cell densities than other methods. Depending on the cell type, it can be anywhere from maybe 1 to 2 times 10 to the 8th per mil.
Next, can I use it for insect cell transfection?
Absolutely. So I showed data for GFP plasmid expression from SF9 cells. We've had a variety of clients use SF9, a number of other insect cell lines for protein expression, and they get very good expression levels.
Right.
One of the other benefits of using Maxcyte electroporation is if you eliminate the need to use baculovirus, it actually helps during the purification process because you don't have any of the contaminated baculovirus proteins that you need to get rid of. So that's an additional benefit of being able to use Maxcyte Transient Transfection over a baculovirus system.
Okay, and another question related back to the plasmids. Do you use linear or circular plasmids?
We tend to use linear plasmids for stable cell line generation.
It looks like we've covered all of the questions. So I'd like to thank our presenter today, Dr. Krista Stager. I'd like to thank Maxcyte for sponsoring the webinar today. And most of all, I'd like to thank those of you who have taken the time to come and listen. We hope you've gotten some information that will help make your research a little bit easier. So with that, I'll say thank you so much and have a great day.